So now that we've covered mitosis and meiosis, we can talk about gametogenesis, or the forming of eggs and sperm, the gametes. Now, in overview, the process for eggs and sperm is actually quite different, although the cell cycle portion of it remains relatively similar. Reminder that gametes are the only human cells to undergo meiosis. Okay, so that's a special cell cycle just for them. And the cells that give rise to the human gametes originate very early in embryogenesis. Okay, spermatogenesis, the process of forming sperm, begins after a boy goes through puberty and then continues throughout his adult life. Okay, theoretically into his 70s, 80s, or 90s. Um, and a reminder that when you go through meiosis, you start with one spermatogonia, okay, or a sort of a preliminary sperm cell, and you get 4 haploid spermatozoa or mature sperm out of it. Whereas oogenesis is going to begin during the fetal period, but arrest partway through meiosis, and then pick it up again at puberty. Now, oogenesis ends at menopause, so women do not remain fertile throughout their adult life. And then just a reminder that when you go through oogenesis, you only get 1 oocyte, not 4. Okay, so for both eggs and sperm, men and women, okay, the precursors to the gametes are the primordial germ cells. These are going to be the start of the germ line, okay, and they arise in the yolk sac, which is a support structure that sort of comes off of the early fetal belly. They arise in the yolk sac during gastrulation, which we will cover in later lectures. But they migrate out of the yolk sac and along what's called the gut tube, so the preliminary gut, to the dorsal body wall, so the back. And then they stop on either side of the developing kidney tissue. So they aren't kidneys yet, but they will be. And when they settle there, their presence induces the development of what are called the genital ridges, which will ultimately become the gonads being the organs that produce eggs or sperm. Now, we're going to go over this migration in greater detail in later sections, but this is the basic idea. Now, while the primordial germ cells are migrating, they do multiply via mitosis, so they continue to replicate and increase in number. And they're not yet going through meiosis. This is mitosis. And in reality, occasionally, primordial germ cells will settle in areas other than the genital ridges, and on occasion, instead of degrading, which is what they usually do when they end up in the wrong spot, they can continue to grow and develop into what's called a teratoma. Okay, so this is just showing the yolk sac, and these little black dots would be the primordial germ cells migrating, and then they end up at the genital ridges. Now, a teratoma is a tumor, and it's a tumor derived from primordial germ cells, which makes it a very interesting tumor. These tumors can contain tissues from all 3 germ layers, and we'll talk about what those are later on, but the idea being that they cover lots of different tissue types, and they're pluripotent, meaning that they can develop into many different kinds of cells or organ structures, which creates the interesting phenomenon where a teratoma can contain actual body structures. So they've been found to contain teeth, hair, functioning glands, etc. Now, the most common tumor in newborns is a sacrococcygeal teratoma. Sacrococcygeal just describes its location. So your sacrum and your coccyx are the base of your spine or your tailbone. So these are teratomas that form at the base of the spine or on the rear. They can be seen prenatally, and they can be a serious complication because the larger they are, the more stress they put on a baby's system and can actually increase the risk for miscarriage or stillbirth. Now, they are treatable. They can be treated surgically, and sometimes even before birth. If you're interested in learning more about that, I do highly recommend the CHOP website, the Children's Hospital of Philadelphia. It's one of the few centers in the country that does this sort of prenatal surgery, and if you want to learn more about it, I recommend you go and have a look. But the prenatal surgeries are possible generally only when the teratoma is actually stressing the baby's system to the point where the baby's life is in danger. It is very rare for these tumors to be malignant. So these are usually not cancerous tumors, just tumors, abnormal growth of cells. There are lots of different types. They can have both internal and external components, and certainly the larger they are, as we said, but also the more they are inside the baby's body, the more complicated they become to treat. And that's why they're treated at specialized centers. Now, I had mentioned that they can be detected prenatally, and I wanted to give you an idea of what that looks like. So just to orient you in this picture, this is an ultrasound image of a baby. On the left side, that sort of thick band of gray tissue, that's the mother, okay, the wall of the uterus most likely. And then you sort of see the baby's head, and you're looking at the baby in profile, so that bright arc-shaped line would be the bone of the baby's forehead, and then you progress along the baby's belly, and then you see the leg, one leg, which is bent at the knee, and at the end you get this big mass, and that's the teratoma. So certainly the larger ones are quite easy to spot by ultrasound, and babies that have them get a lot of monitoring. Okay, so on to spermatogenesis. How do we make sperm? First, a quick review of male function and male anatomy. So remember, sperm production begins at puberty. Okay, so this is not something that starts early in fetal life. And it occurs in the seminiferous tubules of the testes. Okay, so there, for anyone who needs a quick refresher on male anatomy. Now, testosterone production increases with puberty, and that's actually what triggers this process to start. Okay, so these are the seminiferous tubules highlighted here within the testicle. Okay, and these PGCs, the primordial germ cells, are now called spermatogonia, indicating that they are now ready to start forming sperm. Okay, and within the seminiferous tubules are cells called Sertoli cells, and they are going to support the developing sperm. They protect them, they provide resources. Now, initially, the spermatogonia, so these very early sperm cells, are attached to the Sertoli cells via what are called these tubulobulbar complexes. And they serve an important function because sperm aren't supposed to have lots and lots of cytoplasm. Sperm are race cars, they're streamlined. So they offload their cytoplasm through these complexes into the Sertoli cells. Now, sperm, when they go through their development, go through a number of different stages. So once they begin DNA replication, they become primary spermatocytes. As they progress through meiosis 1 and finish meiosis 1, they're now secondary spermatocytes. And then once they complete meiosis 2, they become spermatids. Now at this stage, the spermatids are actually all joined, so the 4 spermatids from the one original spermatogonia are joined by cytoplasmic bridges. But at spermiation, they separate from one another, mature into spermatozoa, or sperm cells as we know them. They break apart from both each other and the Sertoli cells, and they're released into the seminiferous tubules. Now, sperm structure is fairly simple. They have a head which contains the nucleus and the acrosome. The nucleus, of course, contains a haploid number of chromosomes. The acrosome is important because it contains enzymes that, once released, will help penetrate the egg. Then there's the mid-piece that contains mitochondria, and that's really what fuels the swimming motion of the sperm, and the rest is the tail. Now, sperm can be abnormal in any of a number of very interesting ways. They can have multiple heads, multiple tails, they can be abnormally structured, they can swim in circles, and if you're interested in the abnormalities of sperm, you should definitely have a look at a lot of the infertility resources. They'll give you an idea of what's considered normal versus abnormal for sperm. Now, once the sperm are made, they're stored in the epididymis, which is this structure coming immediately off of the testicle, okay, and then it leads into the vas deferens. So at ejaculation, the sperm are going to pass out of the epididymis through the vas deferens. They're going to mix with a bunch of other secretions that from several glands in the male anatomy, and that sort of mix there is called semen. And one ejaculation can contain up to 300 million sperm. So men make lots and lots and lots of sperm because very few of them are actually going to get to the egg. Sperm are capable of fertilizing an egg for about 3 days within the female system, so they can survive for a little while. Important thing to keep in mind if you're attempting to prevent a pregnancy pregnancy. Um, and in fact, capacitation is the idea that once the sperm are in the female reproductive tract, that acrosome that contains these enzymes that are going to help to break down the protective layer around the egg, that it becomes ready. So this capacitation process makes the acrosome ready to penetrate the egg, and that's triggered by entry into the female reproductive tract. Okay. Now, in contrast with oogenesis, there is a portion of this process that occurs during the fetal period, and then it pauses and begins at puberty. So during the fetal period, we make ovaries and oogenesis begins, but the primary oocytes then arrest partway through meiosis, which they pick up again at puberty, which for most women begins around age 11 through age 14. The menstrual cycle, of course, lasts roughly 28 days, although it varies from woman to woman. And during that time, one oocyte is going to mature and complete meiosis. The uterine endometrium, which is the lining of the uterus, is going to grow and fill the uterine cavity, creating a place where a fertilized egg could form a home. Ovulation will occur, so one oocyte is going to be released. And if it were to be fertilized, then the fertilized egg would implant in that lining. If it's not fertilized, then it's shed. Okay? And for women, their fertile period usually ends between the ages of 45 and 50 with menopause, unlike men who remain fertile for the remainder of their life. Okay, so in more detail, the process starts the same way that it does for sperm. The primordial germ cells migrate out of the yolk sac, they reach the genital ridges, they differentiate into what are called oogonia. Okay. At 12 weeks gestation within the fetus, the oogonia begin meiosis, but they arrest fairly shortly after they start in prophase 1, and now they're called primary oocytes. During that time, each primary oocyte nucleus is going to fill with fluid. And that fluid is, in theory, protecting the DNA because it's going to be years, a decade or more, before meiosis is going to complete. So this formation of filling the nucleus with fluid, called the germinal vesicle, is a protective effort to save the DNA from degradation during the time in which meiosis is arrested. Now, additionally, those primary oocytes get enclosed in a capsule, so the cells surrounding the primary oocyte sort of coalesce around it, and the structure of the oocyte plus the capsule are called the primordial follicle, and that's another protective mechanism. By 5 months gestation, all of the primordial follicles are formed and complete. There are roughly 7 million of them, although most will degrade, and by the time a girl gets to puberty, you only have about 400,000 primordial follicles. And so that's the future number of eggs that you can release. And then you get to that point and everything stops until you get to puberty. Okay, again, this is for women. So what does the cycle look like at puberty? So in the blue box, I've shown what happens during the fetal period, and then we pick up again with our primary follicle once puberty begins. So with each menstrual cycle, 5 to 12 follicles are going to reanimate. Okay, so it's not one, although by the end we'll just have one. And once they're sort of reactivated and ready to go, they're called primary follicles. Around the primary follicle forms this layer called the zona pellucida. It's a protein layer, and basically it's a protective coating around the oocyte that will ultimately serve as a barrier to sperm. Additionally, the cells within the follicle begin to grow and enlarge the capsule. So the follicle grows and it's getting bigger. Now, as this process progresses, some of those follicles that initially reanimated are going to degrade, and so until ultimately we really just have one. But the ones that survive up to this point start to take on fluid, and they create this fluid-filled space within the capsule, okay, within the cells that have formed and grown around from the capsule, and that space is called the antrum. And ultimately what's going to happen is the oocyte is going to move into the antrum as all the one surviving oocyte after all the others have degraded. Additionally, during this time, the tissue in the ovary surrounding the primary follicle begins to vascularize. It acquires blood vessels that are helping to support this process. Okay, so during ovulation, continued by day 13 to 14, we're starting to see a surge in two particular hormones, the follicle stimulating hormone and the luteinizing hormone. Okay, and these are going to trigger the continued process of ovulation. So the germinal vesicle, okay, that was our enlarged nucleus swollen with fluid protecting the DNA, starts to break down, and that allows the primary oocyte to pick up with meiosis. Okay, and as meiosis 1 completes, the primary oocyte becomes a secondary oocyte and a polar body, which is essentially just a little packet of chromosomes, the chromosomes that the ultimate future egg is not going to need, remembering that we're not going to make 4 separate gametes for oogenesis, just the one. Now, the secondary oocyte completes this process of moving into the antrum, and the follicle sort of works its way towards the surface of the ovary in preparation for rupture, where it's going to be released into the oviduct. Now, within the oviduct, which is the tube that's sort of adjacent to the ovary that allows the egg to pass into the uterus, it's lined with cilia, and they're going to sort of push the egg along. Now, an oocyte within the female system can be fertilized for up to 24 hours after it's ovulated. So again, there is a little bit of wiggle room there for conception. Okay, now the follicle that the egg sort of hatched out of when it was released into the oviduct is now called the corpus luteum, and it's still important because it's secreting hormones, estrogen, that are going to promote development of the uterine lining and its continued growth and maintenance. So they sort of keep that lining ready for potentially a future baby. If no embryo ends up implanting, then the corpus luteum degrades, the hormones are lost, and the uterine lining is passed. But in fact, there are women who have a history of early miscarriage who are found to be lacking in progesterone, which is one of those hormones that helps maintain and support the endometrial lining. And so by giving them progesterone supplementation, you may make it possible for them to carry a pregnancy past this first trimester stage. The picture that I'm showing you here, this is an ultrasound image, and it's an ultrasound image with color, and the color is highlighting blood flow. So the dark space in the middle, that's going to be your corpus luteum, with the blue and red around it highlighting the vessels that have formed around it to support the oocyte as it developed. So just in summary, to sort of wrap up the differences in the timeline here, for spermatogenesis, the formation of sperm, this period of the primordial germ cell migrating into the genital ridges and getting to where it belongs, that happens weeks 4 through 12 of embryonic development. But everything else, the entire process of meiosis, happens after puberty. Whereas for women, we actually get through prophase 1 of meiosis 1 during weeks 4 through 12 of embryonic development, and then we arrest until puberty and continue the process. Okay, and then a reminder that one of the reasons that we see increased rates of fetal aneuploidies like Down syndrome as women get older is because the oocytes have been arrested in meiosis for longer and longer periods of time, and the mechanism that's maintaining the chromosomes during that time gets older, and it becomes more likely to fail, and we're more likely to see abnormal divisions of chromosomes between cells.